What's going on, folks? I don't know if anyone's in here or not, but this is Unit 3 review, and if you got questions, hopefully I can help. Hopefully. Don't know if anyone's in here or if I'm even live yet, but it looks like I am. What's going on? I'm actually went to Khan Academy to pull up a question or two. Someone asked if I would do some questions from there. So all right, we got some folks on. I can see now. So yeah, if you guys have questions, ask, and yeah, we'll get started. Test is a similar format that we're used to. Act like the electron and explain what is happening. I can do part of that. I probably won't act like an electron because I don't know what an electron acts like. But I will definitely explain that whole process for sure, and just start at the beginning and kind of go through that full show. All right, this is really important. Seeing these two things is linked. Okay, so yeah, I can do that. So seeing these two things is linked, very important. In fact, in many ways, they are opposite of one another. And so, and we'll kind of go through that as we're talking about it. You got energy coming in, right? And you've got CO2, H2O going in, and that makes glucose and oxygen. And then over here, you've got glucose and oxygen going in, and you've got energy going out, right? And these two things just kind of continue on. And I'm assuming these arrows probably are representative of heat. And so that's a really big idea, right? And then I don't think I put those reactions on here, no. And then we talked about even kind of smaller, big idea, right? This is photosynthesis and how H2O, CO2 in, O2 glucose out. And you kind of have this like coupling that's going on here with NADPH and ATP giving up energy, turning back to NADP plus ADP, and so forth, right? And then you have this kind of coupling as well, where you have glycolysis is breaking down glucose, and then it's basically undoing everything that photosynthesis did. Electron carriers are picking up, and they're putting that energy into ATP. And so ATP is the output, whereas here, light is the input. Now, that's all big picture, right? So we'll get down in the weeds a little bit here. So we have photosynthesis, light dependent, and Calvin cycle. Big idea with light dependent. What's the purpose of light dependent? Is to make these two things, ATP and NADPH. And notice it's even like the same colors. It's like yellow, orange, yellow, orange. It's kind of like, I think of it as like energy is being transferred, right? And transformed, even changed from light energy to chemical energy, and transformed from the light to ATP and NADPH, right? And so that is the purpose of the light reaction. Oxygen's made, but the purpose is to make ATP and NADPH. And the purpose of the Calvin cycle, I'll take out one of my headphones because I feel like I'm talking in a vacuum or something. The purpose of the Calvin cycle is to fix CO2 to make glucose. Remember, carbon fixation is fixing or taking unusable carbon and making it into glucose. How does it do that? It takes the energy from the sun, which in this form is ATP and ADPH, and putting it into the Calvin cycle. Those things are kind of like unburdened, or not unburdened. What's the right word I'm trying to think of? Emptied is the right word that I'm thinking of. And they go back to the light-dependent reaction, and that whole thing kind of continues, right? So what are the details of that? Well, here's the light-dependent reaction. I wrote down some things. Let's look at these things that I wrote down. We're not going to look at those things that were wrote down. All right, so the question was, pretend I'm an electron. And so that's what I'm going to do. I'm going to pretend I'm an electron. And so here's photosystem 2 and photosystem 1. These are groups of pigments, right? And in these groups of pigments, you have chlorophyll. Chlorophyll has magnesium. Magnesium has 2 valence electrons. And so when light is basically kind of funneled into chlorophyll, right, and it causes those valence electrons to be excited, and then they leave, and they go into this area right here, which is called the ETC, which I don't have written here. What happened? No. I just switched to like this thing here. And this is supposed to be the highlighter. Sorry. I was going to try to highlight something, but whatever. I'm not going to do that now. So they go into the ETC. We're going to leave that alone. This area right here is ETC for now. Okay. And so how do you keep a steady stream of those electrons going? Well, this is where water comes in. So water reacts here at photosystem 2, and it's split up. And so what you see down here is a single water molecule is being broken up into an oxygen, 2 hydrogens, and this should be 2. There should be a 2 right here. I'm afraid to click up here because I know what's going to happen. It's going to go away again. But these are 2 electrons. To replace the 2 electrons that magnesium lost, right? It's like this constant stream of electrons. And so I am this electron, and I'm just traveling through the ETC. Now, what is that doing? Well, as I travel through the ETC, you've got this series of little proteins in here. They have names. You don't need to know their names. And one of these is a proton pump. And as I just explained this to my honors students, what do proton pumps do? They only have one trick. They pump protons, right? And it's a pump, so it needs energy. And so the energy is going to come from this electron. Why does that electron have energy? Well, it just got hit with light, and so it's energized. It's going to pump protons from an area of low concentration up here in the stroma to an area of high concentration, which is in the thylakoid lumen, which is inside the thylakoid, right, which is inside the chloroplast. And so if we're going low to high, right, that is active transport, right? And so you have this concentration gradient building up inside the thylakoid. I'm still the electron, and so I turned on this pump, caused it to work, and now I'm getting tired, so I'm going to go to photosystem 1 here. I'm going to get re-energized by light, and then here comes NADP+. It's going to scoop me up, and it becomes NADPH, and NADPH is an electron carrier, and it's going to carry me off to the Calvin cycle. And so that's where my story ends for now. But we do have to talk about this concentration gradient that we built up. So we have a concentration gradient of thylakoid, or of hydrogen ions or protons inside the thylakoid lumen. And that gradient is going to be used by this protein called ATP synthase. What does ATP synthase do? It makes ATP. How does it do that? It goes from area of high concentration to low concentration, diffuses, so that's called facilitated diffusion, causes this little thing to spin around, and that mechanical kind of energy causes this phosphate to be put onto this ADP to make ATP. ATP is the cell's energy currency. You got ATP, you got NADPH. Those 2 things represent the energy transfer from light. Where are they going? They're going to the Calvin cycle. Let's see. Okay, yeah, this word right here. This is a special little word, chemiosmosis. All right. It is proton gradient is used to create ATP in this process called basically that diffusion of high concentration to low concentration using ATP synthase is just this special word. Just think chemical. except for the no CHCAL on the end, just chemical osmosis. I don't like that word because I think it's just weird. It's extra. But it is a word that you'll see in AP literature a lot. So make sure that you are familiar with that word. And it is literally just using that concentration gradient to make ATP. Okay. That's light dependent. Moving on. So here's the Calvin cycle. And so the purpose of the Calvin cycle is to fix carbon, right? Take unusable CO2, make it into usable something else. And here are those things we made in light dependent, right? ATP, NADPH. They're going to be used here. We're going to talk about them in a second. So here's our CO2. Now, technically, each CO2 is going to get on separately and kind of make the round. That I was asked a question today, like how many rounds or how many Calvin cycles does it actually take to make a glucose? 6. It doesn't really matter, right? And so this picture kind of shows it just taking twice. Literally doesn't matter. As long as you understand that these 3 CO2s get on. How do they get on? They get on with this enzyme called ribisco. Ribisco adds them to these REBPs, makes some 6 carbon molecules that have a name that nobody remembers, and that splits in half, and you have these PGAs. And these PGAs are what ATP and NADPH are going to kind of apply themselves to, for lack of a better term, right? So you have a phosphate here, you have some electrons here, and what ends up happening is the product is this thing down here called G3P. And so NADPH adds its electrons, becomes NADP+, goes back to light dependent. ATP goes adds a phosphate, goes back, becomes ADP, goes back to light dependent. And you have these things called G3Ps. You need 2 G3Ps to make a glucose, right? And then this whole system is recycled again to start over the Calvin cycle, which isn't that important for us. Calvin cycle is actually like 2 sentences as far as AP is concerned. So big ideas here is carbon fixation. These molecules being used to change this PGA into a G3P, and 2 of those G3Ps equals a glucose. Now, the question was, pretend I'm an electron. Right now, I am part of G3P. All right. So I went from magnesium/water through the electron transport chain, photosystem 2, photosystem 1, NADPH, added to a G3P. So now I'm part of a G3P, and I'm actually part of a glucose because 2 G3Ps come together. That is Calvin cycle. Let's make all this work on this picture, right? This isn't the same picture that I gave you, but it's close enough, right? So here's that same thing, right? This is just kind of going over and over. It's H2O, CO2 are in, O2 is the byproduct, and glucose comes out. Cellular respiration. All right, so let's get a big picture. Here's the big picture. So big picture is we have our glucose. That goes in, and what comes out? ATP. That's it. There's a lot of things going on in there in between. But we're going to worry about big picture, which is glucose goes in, ATP goes out. Now there are 4 phases of cellular respiration. Glycolysis is first, glucose to pyruvate. Pyruvate oxidation is second. It's just kind of takes the pyruvate, breaks off a carbon, gets it ready for the Krebs cycle, because the Krebs cycle only takes in 2 carbons. And I'll show you a picture of pyruvate oxidation. It'll make a little more sense. Nothing specific about redox reactions. I can go back up here and show you, though. Basically, what's going on here is a redox, right? When electrons are added to something, that something is reduced. What does that mean? Its charge is reduced, right? And because it's added electrons to it. When something gives away electrons, it's been oxidized. What does that mean? I don't really know. Like I couldn't tell you why that term is used. It probably has to do something with oxygen being the thing that takes electrons. That would be a Mr. Crow question. But Basically, think of redox reactions as the accepting and giving away of those electrons, and that causes that exchange of energy. Yeah, we'll talk about acetyl-CoA when we get back to here. Okay. And then those two carbons get on the Krebs cycle, and Krebs cycle makes electron carriers. Those electron carriers go into the electron transport chain, and those electron carriers drop off their electrons, which are accepted by oxygen, and ATP is made. That's the big picture for cellular respiration. Let's talk about the individual steps. And I'm going to talk about the individual steps on this one because they're all here. So here's glycolysis, glycolysis, breaking down glucose. Here's glucose. Goes through glycolysis. You've got some ATP that's made. You got some NADH that's made. And you got 2 pyruvates that are made. What's interesting about glycolysis, if I could show you a picture actually. I don't know if this will be helpful to you or not. You don't need to know this, but it could be that it's helpful to you for some of you to kind of like wrap your head and say, oh, okay, that makes sense. Again, I don't know if this will be helpful or not. All right. So this is glycolysis a little more detailed, but it's still glucose, pyruvate, NADH, ATP, right? So don't get too confused. But remember back in Calvin cycle, I talked about how these were opposites, and so I think that's why this is important to me. Calvin cycle, we had to take 2 G3Ps to make a glucose, right? Well, here, if I split this glucose, what should I get? If it's just the opposite, I should get 2 G3Ps, right? That's exactly what this is, right? We just kind of skipped over this whole thing, but this is a G3P, this glyceraldehyde 3-phosphate. That's what G3P stands for, which you don't need to know, and don't worry about all this over here. But it goes through some more things, and then you get a pyruvate. And so I think that just showing you this is literally an undoing of Calvin cycle will maybe help some of you. I don't know. All right, so at the end of this, we got some pyruvate, and that's the important thing. Let's talk about pyruvate oxidation. So here's pyruvate over here. All right, here's pyruvate. Pyruvate, and this is a mitochondria, cytoplasm. So pyruvate goes into the mitochondria, and when it does, it's going to be oxidized, which is going to give away some electrons. When it does that, well, if it's giving away electrons, look what comes to get them, NADH, which is an electron carrier, right? It's going to drop a CO2, so if it drops a CO2, that means it drops a carbon, so it's going to go from 3 carbons to just 2 carbons. And at that point, when it becomes this 2-carbon thing, there's this enzyme called coenzyme A that's going to come in and scoop up those 2 carbons, and it's going to carry them to the Krebs cycle. Think of it as like an escort. If it's the undoing of the, well, it's kind of like, because the making of glucose is its own end, because plants can use glucose and plants can use just G3P. For that matter, just to do other things. These two, let me go back up here. You can think of it as these 2 reactions kind of co-evolved together to kind of coexist. One of them undoes the other ones, and notice, but they kind of feed into each other, and so they're kind of, and plants are able to do both of them. I don't know if that answers your question or not, but if you didn't undo it, you just have a pile of glucose with nothing to do with it. All right, and so those 2 carbons are going to be escorted to the Krebs cycle, and this whole thing, coenzyme A plus these 2 carbons, is called acetyl-CoA. You don't see it in the literature anymore. It used to be a really big deal. It's still a big deal, but we just don't talk about it as much. But if that helps, that's fine. That's pyruvate oxidation. It's literally giving away an electron. When it does that, electron carriers come scoop those electrons up, loses CO2 or loses a carbon as a CO2. Got 2 carbons left. They're going to be put on the Krebs cycle. What's Krebs? We're not going to go to that picture. So the purpose of Krebs is to make electron carriers. Here's pyruvate oxidation right here. 3 carbons, loses CO2. Electron carriers come pick up electrons. Coenzyme A picks up these 2 carbons and it's going to escort them to the Krebs cycle. Plops them right on there. And lo and behold, those 2 carbons are going to be lost too. Guess what? As CO2. So when you exhale, that CO2 in your breath is product of the Krebs cycle. And because these CO2s are all these carbons are all kind of broken up, again, undoing of the Calvin cycle. Here, electrons were added, right? In order to make this. Here, this is taken apart and electrons are loosed, and the NADH is going to come pick those electrons up. Also, FADH2, and an ATP is made, which is not that important. I mean, that ATP is not unimportant, but it's not important for us. The big purpose of the Krebs cycle, lots of NADH is made, right? And of course, Krebs has to happen twice for each glucose molecule because there's 2 pyruvates. Where are those electrons now? Speaking of those electrons, they're being carried by NADH or FADH2. And then lastly, ETC. So ETC is kind of the opposite of light dependent, right? Because let's think about light dependent. Light dependent was you had water coming in, being split up, oxygen given off as a byproduct, electrons going through here and eventually being scooped up by electron carriers, right? Here you have electron carriers dropping off electrons. Oxygen is not a byproduct, but is a necessary reactant, and rather than being the first reactant, water is the last product here. So it literally is the exact opposite. So what does this mean? Well, here's these 2 electrons again. They're being dropped off, and these blobs here that are labeled 1, 2, 3 are proton pumps. What do proton pumps do? They pump protons. Here you have an area of low concentration of protons high up here. They're being pumped from low to high. It's active transport into this intermembrane space, building a concentration gradient. And as these electrons are going to move along these little protons, those redox reactions again, if you just knew their name, that'd be sufficient. If you didn't know their name, that would also be sufficient. But these electrons have energy, and what is that energy being used to do? Turn on these proton pumps. What do? They pump protons. 1, 2, 3 protons are being pumped across, and those protons are going to build up in this intermembrane space, which is going to be used, that chemiosmosis again, ATP synthase, which makes ATP. And remember how I told you it would be really easy to get this mixed up with this, right? They even kind of look the same, except one has greens and purples, and this one just has nothing, other colors, right? And so don't get them mixed up. Don't make some photo respiration synthesis mutt thing. Make sure that you keep them separate completely. They're opposites of each other. There's a lot of the same kind of thing, but just keep them separate. And so those electrons turn on these pumps. There's oxygen. What does oxygen do? It's the final electron acceptor. When this last protein gives that oxygen away, oxygen accepts it. You take 2 electrons, an oxygen, and 2 protons. You get H2O. And FADH2, we won't forget to talk about him, even though a lot of people do. He drops off his electrons here, and they only turns on 2 pumps, and so it's only 2 protons, which is only 2 ATP. I don't think I have a totals thing. You don't need to know totals. Just it's 36 to 38. One more thing, and then I'll stop and ask for clarifications if you have them. All right, fermentation. What is fermentation? Fermentation is a process that only occurs if there's no oxygen. If there's oxygen, first of all, what is this? This is glycolysis, right? Glycolysis doesn't need oxygen. It happens in all living things. And if there is oxygen, well, then you've got acetyl-CoA here. That's that pyruvate oxidation. You got Krebs. You got ETC happening here. Everything's good to go, right? Because if there's no oxygen, think about it this way. If there's no oxygen, where is NADH going to drop off its electrons? It can't, because there's nothing here to finally accept them. If NADH is unable to drop off its electrons, there's no NAD+, right? If there's no NAD+, none of this even happens. Glycolysis can't even happen, right? Because it's a reactant of this whole thing. And so if there's no NAD+, glycolysis can't occur. Enter fermentation. If there's no oxygen, then there's this other process. The purpose of fermentation, well, I've got it written down here in no uncertain terms. Occurs so that glycolysis can continue. It frees electron carriers, creates NAD plus from NADH. Again, here's glycolysis, glucose to pyruvate, NADH, ATP. But if there's no oxygen, NADH has to drop its electrons somewhere, and so it drops them off in pyruvate, essentially, whatever. Turns pyruvate into something else, so there's a product there, and that NAD plus goes back to glycolysis, and this is kind of a way to kind of keep glycolysis going. That's why this is called NAD plus regeneration. Different kinds of organisms do different kinds of fermentation. Obviously, plants don't need to do fermentation because, well, they make their own oxygen. But we do that. This is actually animal fermentation here. Bacteria, yeast do alcohol fermentation, which produces CO2 and ethanol. We didn't really talk about the differences. AP doesn't talk about the differences. Big ideas here for fermentation. And that is all of photosynthesis, cellular respiration in a very quick blip. Any questions about any of that? Any thoughts? Any concerns? I don't think I missed any questions. No, don't think I did. It's funny, I just taught all this to honors too, so it's really fresh. Well, the first 3 sections of this unit were about enzymes, and so don't forget them. Enzymes aren't too bad. It's not too bad, right? As long as you just remember your terms, enzyme, substrate, active site, all those terms, right? The enzyme is unchanged. The substrate is the thing that's being changed. You got this concept of induced fit we talked about, naming. Okay, and then graphs like this, being able to look at a graph. There was a good question one of you guys showed me in class, and it had reaction rate here, it had substrate concentration, and this was on the bottom. It was numbered like 1, 2, 3, 4, 5, 6, 7, and it said from the data, what can you expect to happen between minutes 7 and 10, which 10 was kind of a minute that wasn't on the graph. It's basically asking you, if you keep going, do you expect the rate of reaction to continue? If you increase substrate concentration, do you expect the rate of reaction to continue? No, because it's already flatlined. Basically, it's already like saturated, right? The enzyme can't work any faster. And so being able to look at a graph like this and answer questions about it is going to be pretty important. Again, I haven't even looked at the test yet, so answer specific questions about it. But I know you guys probably don't like that, but you know what? That's going to be all right. Yeah, and 2 different kinds of enzymes. These are just, some enzymes build things, some of them break things. Yep, I can do that. Yeah, fitness is real simple. It's a real simple concept. All right, so this word, fitness, is an evolutionary concept, first and foremost. So we're talking about things that change survival, right? And so that's why you have the picture of the birds and this ability to survive and reproduce. and I gave examples of, think of this as like molecular fitness, right? So this is a picture that's got like 7 different chlorophyll molecules on it. Why would a plant need 7 different chlorophyll molecules on it? Well, let's take 2 plants that are competing for sunlight or whatever in a forest. One of them has 7 different chlorophylls on it, the other one has 3. Which one is going to be able to take in more different kinds of light? The one that has 7. What do I mean by different kinds of light? Well, there's light of different wavelengths, there's different colors, there's all this different things that are going on with light, right? And so the idea here is that the one that has more variety is going to be more fit because it's going to be able to better adapt to its environment. It's the whole beak thing, right? So that's it. And then like I gave an example of cholesterol. Variations in cholesterol. Cholesterol is a steroid that's in your cell membranes that gives it fluidity. And variations in the amount of cholesterol in cell membranes is going to allow you to live in different kinds of environments. And then we talked about hemoglobin, talked about the 2 different kinds of hemoglobin subunits, how fetuses have gamma subunits, which attract oxygen more than adult beta subunits, which allows the fetus to actually pull oxygen off of the mother's cells or hemoglobins, which gives them a better rate of survival. So the whole idea here is very fast. The idea here is that if there's more variety in the molecular realm of things, then that organism is going to have a better chance to survive. I'll give you an example of something too that's interesting that I didn't talk about in class, but I just thought about. I think it's this little molecule right here, or maybe this one of these 2 little proteins is called cytochrome C. And cytochrome C is a protein that all living things have. You also find it in, it's one of these 2, maybe it's this big one here, or maybe it's one of these. I don't remember exactly which one it is. It doesn't really matter. But it's another example of fitness. It's something that works. It's something that does a job well, and you see it. You find it in all organisms because it is something that does its job well. So that'd be anything. Basically, this unit or this section is this unit's nod to evolution. Show a coincidentally similar FRQ and work through it, or give a hint. Okay. So yeah, like what sort of FRQs might you see? Well, I posted one, and I had to be careful. You know, I don't want to get myself into trouble talking about FRQ released questions or anything like that. But I posted one that kind of talked about products and reactants of cellular respiration and photosynthesis. And literally, if you knew this equation, you could kind of work through it, right? And so that's the thing, is I think when students come to this stuff, they get so bogged down. You've heard me say this so much, you're probably sick of hearing me saying it. But you guys get so bogged down in like this minutia that you'll just forget this. You know, you'll just forget like this. You'll forget that water goes in and doesn't come out. You know, just something simple like that. And so I think that's the big thing. Get your test, write down this equation, you know, just to kind of give you some grounding, and then help you stay grounded. This is photosynthesis. The reverse is cellular respiration. I know that's probably not as helpful as you were hoping, but you know, you could get a graph, and the graph would. I haven't seen the test, so I'm just. But you know, you're thinking about preparing for the AP exam in May too, obviously. And so, just knowing. So if you look at this picture, what's going to affect the rate of reaction in photosynthesis? Well, the amount of sunlight, the amount of water, the amount of CO2, right? All those things are going to directly affect the rate because they're all inputs, right? Same for cellular respiration. What's going to affect the rate of cellular respiration? The amount of glucose, the amount of oxygen. And so just think about things that way. Don't get bogged down in the details so much. So if an exergonic reaction comes before an endergonic reaction in a pathway, the energy of the first reaction can be used to power the second, okay, good question. And I think, let's just use this. Okay. So going from ATP to ADP is an exergonic reaction, meaning that after that happens, there's energy that has been used, right? ATP goes and phosphorylates something, turns on some sort of process, right? And there's less energy in the products than there is in the reactants, right? That's an exergonic reaction. So if it comes first, right, the energy of the first reaction will be used to power the second. Sure, you could theoretically use the energy from ATP, or that, you could potentially use the energy of ATP being changed to ADP to change ADP back to ATP. However, what's the problem? There's a loss of energy here, right? And I wish I could draw an arrow. There's a loss of energy here as heat. That is entropy. This is not a perfect cycle, right? We could take literally any one. We could take this one, right? You make this glucose, you make this glucose. Is there enough energy in this glucose to make enough ATP to make enough glucose to kind of have this perfect circle? No, you can't, because there's heat being lost all the time. And so there has to be an input from some outside source, which is the sun. Yeah, exactly. Entropy. That's exactly right. There has to be an input from an outside source, and that's the sun. If the sun runs out, well, then we'll be toast. Actually, we won't be toast. We'll be ice. We'll be frozen. But we don't have to worry about the sun running out anytime soon, I don't think. It's funny when I say stuff like that. But when is that going to happen, Mr. Chipman? Don't be afraid. The sun isn't going to run out in your lifetime, or anybody's lifetime, for that matter. It's a long time. I don't know if that was helpful, but yeah. We mentioned enzymes. I'm just going to talk a little bit more about this stuff, inhibitors. Just remember that there's two different kinds of inhibitors, right? And a competitive inhibitor competes directly for the active site. A non-competitive inhibitor binds to some allosteric site, which is not the active site, but it still causes a shape change, which changes the function of the enzyme, right? Anything that changes the shape of the enzyme is going to change its function. And that's why you see these pictures like with pH. There's an optimal pH, but if it gets on either side, it changes the shape. It doesn't function anymore. With temperature, it spikes, and then it denatures, just completely tanks the enzyme activity. And so again, if you see a graph, that's how you need to read that. Graphs are important. Other questions, other thoughts? The long talk on photosynthesis, cellular respiration was kind of all-encompassing. It's literally most of the unit. And enzymes really feed into this other stuff too, since most of those are enzyme type reactions. So, anything else? Oh yeah, I could go find a con question maybe, see if I could pull one up. Do one. Let's see. See if I can find one that's relatively short. They're kind of hard to. Oh, there we go. Oh, I'm glad. I'm glad you thought that was helpful. No, I didn't take it as sarcasm. I typically take people at their at what they're at face value, because I always appreciate it when people take me that way. All right. So here's a question. Literally pulled this off console. Some of you guys have probably already seen it. I have not looked at it, read it. Don't know what the correct answer is, but we're going to look at it together. Because I was asked to go over a question or two. And this is off Khan Academy. This is not my question. This is Khan Academy's question, which are free. And so they probably don't mind me doing this. In fact, I just gave them free advertising. All right. A group of students are conducting an experiment on Elodea canadensis, a species of aquatic plant, in order to test the effect of light intensity. All right, so look, I'm already thinking, when I read that, I'm already thinking about what light intensity does to photosynthesis, right? They place Elodea canadensis plant into an aquarium tank, place each tank at a different distance from the light source. It's almost as if we did this exact same lab. After a few minutes, oxygen bubbles begin to form in each of the tanks. Students count the number of oxygen bubbles released during a 5-minute period from each tank. The results of their experiment are shown below. I think I'm going to do this experiment with the honors class too. I don't want to do the little round discs anymore. All right. So distance between plant and the light source is here. And so I'm just kind of walking you through the way I'm thinking about this question. Right. And so we are measuring light intensity. And we need to think more light equals more photosynthesis, right? And so one of the ways that I can see already that a student would get tripped up by this is they would think more number, more better, right? And you might get it backwards, right? But in fact, the smaller the number is, the shorter the distance between the light and the plant, which is going to be a more intense light, right? And so this is exactly what you'd think you'd see here. As the light is a shorter distance from the plant, there are more bubbles being released, which represents more photosynthesis, right? Because more oxygen is being increased. So before I've even looked at ABCD, I've talked myself through this whole question, and I know what to expect when I go down to ABCD, as opposed to looking at ABCD and then going back up here and trying to find it. That little thing right there is where some of you guys need to kind of park yourself for a little bit. Some of you are just fine. But just, what is this question talking to me about? What am I supposed to see here? And then this question is really open-ended. Which of the following conclusions best supports the results? As light intensity increases, the production of photosynthetic product remains constant. That is not true, because it does not. As it decreases, the production of photosynthetic products decrease. B is the correct answer. I'm not even reading C or D. As light intensity decreases, right, which means that I'm going further away, the product is also going to decrease. I'm not reading C or D because they're not right. If B is right, A, C, and D are all wrong. So hopefully that's helpful. I've done that with you guys, several times. But it's always helpful to kind of hear how someone else thinks about a question and kind of processes it. Yeah, that was right. I just went to con. I don't know if you guys heard that little sound or not. All right, so I'll do one more question here. grab it. I think this is an enzyme question, so it's perfect. Okay, so here we go. And this is interesting because it's an enzyme question, but it's about cytochrome C, which is cellular respiration. So it's kind of like mishing them all together, which is good. Cyanide is a toxin that can affect cellular respiration. It functions by binding to an allosteric site. Okay, all of a sudden my mind is thinking enzymes, right? Allosteric site, I'm thinking non-competitive inhibitor, I'm thinking all those things. Allosteric site on the enzyme cytochrome C oxidase, disrupting the electron transport chain. Okay, so now I have these two things, right? I've got enzyme, allosteric site, disrupting function. I've got ETC, cellular respiration, and that's automatically making me think of concentration gradient, ATP synthase, all those things, right? I'm bringing it all in. Cyanide is able to bind to cytochrome C oxidase whether or not the enzyme is bound to a substrate. Which the following best, and I didn't need any of this, but that's just the way I'm doing it, right? Just the way I'm thinking about it. Which the following terms best describe cyanide. It's a non-competitive inhibitor. That is what I'm going to go with. And how do I know that? Well, allosteric site is my tip-off. But then this last sentence is actually all you need for this. Whether or not it's bound to a substrate, because where does a substrate bind every time? Active site. That would be a competitive inhibitor. This doesn't matter if there's a substrate or not there, so it'd be non-competitive inhibitor. Let's see if I got it right. Ching ching. All right. So yeah. So I don't know if that's helpful. When I sit down and do a question, that's kind of where I'm at. I just think about, I bring it all in, and do that. I don't even know what these other two things, A and C, aren't even things. They're just not things. You just have to be able to tell the difference between competitive and non-competitive. And literally this last sentence tips it off, but I already knew before that. Because allosteric site. All right. Could do one more, I guess. Let's see. I'm kind of interested in it. Okay, this is a graph. Let's do this. I'm going to get one of these wrong. That's what's going to be funny. All right. Consider a reaction in which reactants XY combine to form product Z. The diagram below compares the reaction coordinates. Okay. And so, well, so first of all, what do I know? Reactants, products. I know that B, C represent activation energy, right? And here I see that there's more energy in the products than, or in the reactants than there are in the products. So this is an exergonic reaction. Which of the following statements about letter C is true? This is the activation energy. Letter C represents activation energy. I'm not even, there's a difference. The catalyzed reaction and uncatalyzed reaction. Okay. It does not represent the difference. It is B. It represents the uncatalyzed pathway. What does this catalyzed mean? What is an enzyme? It's just a catalyst, right? And so you can think of it, if it helps you to read it by saying with an enzyme, without an enzyme, that's fine too. But C is the uncatalyzed, because what does a catalyst do? It lowers the activation energy of a reaction. And so the answer is B. All right. I think that's good. I don't know if that's helpful, but just talking through questions, I think, is always a good thing. All right. You guys have any other questions for me before we come to a close this evening? I will be at the school tomorrow, pretty early, probably, because I have to make copies. I actually have to run your test off, and so if you need to come in, my door should be open. Come in and ask questions, or just come in and study, whatever you need to do. But I am going to go ahead and sign off for the night. It's been fun. Y'all have a good